Assalamu alaikum viewers, welcome to my channel Genome Wide Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. So this is the part 2 of database 2 about the long non-coding RNAs. In the part 1 of this database 2, we have described about that how to find the long non-coding RNAs through simple search or browse, and how we can save or extract the 2D structure of long non-coding RNA. So in this part we will discuss about blast analysis and other. So let's start. So when you click on the link provided in the description, so you will come into this page. So in the first video we have discussed about search and browse. In this video we will discuss about the blast. So you will click on blast, and here you can paste your gene sequence, and then here select the clade and family and order as species. uh, Here is the species of your organism, and here you can select either the clade or whatever. So here you select the species and select from this. These are the list of number of species or any. So here you can select paste the sequence of your gene. Here we take the example of that sequence of your genomic sequence, genomic sequence of your gene. So click on it and then run blast. Click on run blast. So in the Zepus Jujuba, so there are 1 2 3 4 5 6 13, about 13 long non-coding RNA was found related to that specific gene. So these are the long non-coding RNA ID in the ALNC database, and this is the species, and these are the score, and these are the E values. And in the below, the alignment of each long non-coding RNA was given. So when you click on any of the long non-coding RNA ID, so it will guide us to that this window. So here is the species, and here is the scaffold of that long non-coding RNA, and this is the sequence of that long non-coding RNA, and this is the dotted structure of that long non-coding RNA that you can copy and paste into your any software to construct the structure. From here you can find out the 2D structure of that long non-coding RNA, and then right click, copy and save it into your destination folder. And here is the length of that, and sequence and this is the GC content, and this is non-coding RNA values or probability values. Here you can click and find out the CDS or peptide sequences. So in this way, through blast search of a specific gene, you can find out the long non-coding RNA related to your specific gene in the species. So by clicking on this icon, so you can save these all information into your destination folder. When you click on it, so you can save it into your destination folder. So this is about the blast. Now we can check the comparison between the long non-coding RNAs and mRNAs in the different species. So click on this icon or search bar. So here you can select your species from this list of species. You can select your species on which you are working. So and then click on visualize. So it will give you the number of microRNA and long non-coding RNA in that species. So this is the dark blue is the number of mRNAs, and this is the light blue is the long non-coding RNA, and this is here is the AU value, where here is the transcripts, that about 5,000 transcripts contain this much long non-coding RNA at zero point 0.5 AU content or zero point 0.6, so 3,143 long non-coding RNA and the 12,186 messenger RNA. So this is the graph. You can also download it from here and then use it into your any manuscript or link to show that how many long non-coding RNA or messenger RNA present in your species. So from here you can download the long non-coding RNA database from your species. So from here you can select the your clade. From here is the order, and then from here family, and from here the species, and then when you click on it, so it will give you the all the long non-coding RNAs present in that species, so you can download them. So click below and here download, and then save it into your destination folder. From more here, if you have find out a long non-coding RNA in your species and you want to submit it in any database, so you can submit it in ALNC database by go to the more and submit data, and then follow the further process. From here you give your name and the email address. From here the publication in which you have published that data, and here some give some information, and then you can submit it. And here is the statistics about the long non-coding RNA present in all the species. Here is the that this is the sample code in which sample, from which sample they have extracted these long non-coding RNA. This is the species name, and this is a clade, this species belongs to which clade, and this is the tissue that these long non-coding RNA extracted from which tissue, and this is the transcripts, RNA sequencing data transcript, this is the ortholog, mate, scaffold, and these all the information about that RNA-seq data. And this is all about that long non-coding RNAs from extracted from this species of that samples. These are the information. So this is all about the ANC database about the long non-coding RNA. Hopefully it will help you. Thank you.